Good evening, everyone. Welcome on this wonderfully, what feels like an incredibly warm evening, even though I'm sure it's only in the 40s, but somehow it seems so much better. I can see a lot of people are squished. There are seats in the middle if people either want to shunt in a little or just sort of ask people, because there are definitely seats, so you shouldn't need to stand. Hopefully people can shunt along and make room for everybody. My name is Jane Pickering. I'm the Executive Director of the Harvard Museums of Science and Culture, which includes the Harvard Museum of Natural History, which is the sponsoring museum for tonight's event, which I'm very much looking forward to. We're going to hear from Hopi Hoekstra, our own Hopi Hoekstra, who is at the Museum of Comparative Zoology, an evolutionary geneticist, and I think it's going to be a wonderful occasion. I would like to just say a couple of sort of housekeeping things. First is, and those of you who come frequently have heard me say it many times, we do have a spring program, which I've forgotten to bring with me, but is actually on a table to my right, and that includes a complete schedule of all our events. And in 2 weeks' time, we actually have Steve Stearns from Yale. He's coming on April 17th to give the final one of this series of lectures, which is the Evolution Matters series. This is the third one, and I would like to take a moment to thank Drs. Herman and Joan Suit, sitting in the audience, who sponsor the Evolution Matters series. And they have been dedicated sponsors of this series for several years, and we all know, or many of us in this room know, how important it is to talk about evolution by natural selection in a public forum, so we're very grateful. Thank you very much. You will have also noticed a new survey on your chairs, and I know some people have already filled it out and handed it in, so if you haven't, I encourage you to do so. It's to enable us to try and really produce public programs that are going to meet everyone's needs and make them better. I'd also like to remind everyone that we do actually have a special session at the end of the lecture with one of Hopi's students, Emily Jacobs-Palmer, on the front row here. And she's doing a special session for teachers, K through 12 or K through 16 teachers, about the topic of tonight's lecture. I think there may be a glass of wine involved. So for those of you who've signed up already, then they'll be meeting just behind the wall here. So you can either come up the stairs through here or go back out of that door and round behind. And if there are any teachers here who didn't sign up, please know you're very welcome to come. So there's always extra spaces. So if you didn't sign up but would like to attend, then please do so. I am going to stop talking now, and I'm delighted to introduce Jim Hankin, who will be presenting our speaker. And Jim is the Alexander Agassiz Professor of Zoology and Director of the Museum of Comparative Zoology here at Harvard, and he's also the Curator of Herpetology, and he is also a Professor of Biology in the Department of Organismic and Evolutionary Biology. Harvard gives professors a lot of names, and I think Jim has some of the most. He is a principal investigator of the Hankin Laboratory, where his team studies the evolution of morphology, developmental biology, and systematics, with a special emphasis on amphibians. So I'd like to bring Jim up to the stage. Thank you.
Thank you, Jane, and good evening, everyone. I actually have one additional announcement, which is to ask those of you, if you have your cell phones, please turn off the ring. I have the distinct honor of introducing tonight's speaker, and I'd like to do so by telling you that it's a great time today to be an evolutionary and comparative biologist. Not only do we continue to discover and document previously unknown biodiversity, new species and the like, but a series of technological breakthroughs in molecular biology, genetics, computer science, digital imaging, and other areas allow contemporary biologists to answer, at long last, many fundamental questions regarding the evolution and diversification of organisms that practitioners of our craft have been asking ever since Darwin, and indeed even before then, but were unable to resolve satisfactorily. In this respect, I can't think of a better person to illustrate the potential, the power, and the discoveries of contemporary biology than tonight's speaker, Dr. Hopi Hoekstra. Hopi is Alexander Agassiz Professor of Zoology in the Museum of Comparative Zoology, where she also serves very ably as the Curator of Mammals. In addition, she holds faculty appointments in the Department of Organismic and Evolutionary Biology and the Department of Molecular and Cellular Biology. Hopi came to Harvard in 2007 from a faculty position at UC San Diego, having earlier completed a bachelor's degree in integrative biology at UC Berkeley, a PhD in zoology at the University of Washington, and a postdoctoral fellowship at the University of Arizona. She has received numerous prestigious awards and appointments. Perhaps the most recent is her appointment, effective just this past January, as a Howard Hughes Medical Institute investigator. I have a strong suspicion that Hopi is one of the few HHMI investigators who maintains an active field program, and I'm virtually certain that she is their only museum curator. The Hoekstra Lab uses wild and laboratory populations of rodents to study the molecular, genetic, and developmental basis of evolutionary change. Their work has been featured widely in the popular press, including the New York Times, Nature magazine, Science magazine, National Geographic, and National Public Radio. Surprisingly, none of those articles discloses what I find to be one of Hopi's most remarkable accomplishments. She is the only biologist I know who has personally taken the rectal temperature of a grizzly bear. I urge you to ask her about this during the question and answer session, which will follow her talk. Ladies and gentlemen, please welcome Professor Hopi Hoekstra, who will now lead us from Darwin to DNA.
Thank you for that introduction, I think. I'm always nervous when Jim introduces me. It's a great pleasure to be here to talk sort of on my home court. It's great to see so many faces, both old friends and new ones. So today what I want to do is tell you a little bit about what my lab has been doing over the last 10 years or so. So as Jim sort of alluded to, much of the work in my lab has been focused on trying to make the connection between genes and traits, and in particular, traits that are important for the fitness of organisms, that is, their survival and reproduction in the wild. Now, most of our early work focused on morphological traits, and in particular, camouflage and coloration, which is nicely illustrated by this image here. So this wonderfully adorable little beach mouse actually lives on the Gulf Coast of Florida. And as you can see from this image, its coat nicely matches or camouflages it in its natural habitats. But in recent years, we've extended that approach to try to make the link between genes and behavior. And the behavior I'm going to tell you about today is also illustrated in this image, and that is burrowing behavior. This mouse is sort of emerging from its freshly excavated burrow. So as Jim mentioned, I'm an evolutionary biologist, so it's hard for me to begin a talk without mentioning Darwin. Since a young age, Darwin was fascinated with beetles, and he had an early hobby, and that was collecting beetles, which quickly became an obsession. So he became, especially in his college days, very competitive with his classmates, including this one who did this now famous image, Albert Ray, and they would compete as how many beetles they collect and the diversity in terms of their taxonomy. But it was clear from Darwin's notes that he wasn't just interested in its taxonomy or the morphology or size and shape of these beetles, but he was absolutely fascinated with their behavior. And he in fact kept copious notes on animal behavior, and this was one of the favorite subjects of his correspondence in his letters. So here's an example of one of the letters with a drawing that was done by one of his sisters. But not only was Darwin fascinated by behavior, he was also slightly troubled by it. So if you look at the origin of species, in chapter 7 he addresses the question, can instincts or behaviors, instinctual behaviors, be acquired and modified through natural selection the same way as he argued that morphological traits were? And he worried that if he couldn't explain how behaviors evolve, this would be a difficulty sufficient enough to overthrow his whole theory. Well, in the same way, he eloquently argued that morphological traits could evolve by natural selection through observation and experiments, and beautifully outlined in arguments with Victorian prose. He concluded at the end of this chapter that just like morphological traits, behaviors could evolve as well. Now, of course, since Darwin, there's been a large number of studies documenting the evolution of behavior. And there are behavioral ecologists who have amassed a large amount of data that support Darwin's conclusion that behaviors can evolve, and largely in the same way. So these days we have a field of researchers who I would call behavioral ecologists, who tend to study a wide diversity of organisms. They tend to study these organisms who display these fantastic behaviors in their natural environments, and tend to focus on questions at the ultimate level, sort of how and why these behaviors evolve. But there's also another group of biologists who study behaviors in a way that Darwin couldn't even imagine. And I'll classify these folks as neurobiologists and geneticists. But their approach is very distinct, yet largely complementary to behavioral ecology, in the sense that neurobiologists and geneticists tend to study or focus on a handful of model organisms ranging from laboratory mice to fruit flies to nematode worms. They study these model organisms in controlled laboratory environments for very good reasons. And the questions are largely fundamentally different. That is, they're more interested in sort of the genetic nuts and bolts of how these changes at the genetic and molecular level actually produce variation in the behaviors we observe. Now, it's this sort of contrast that sparked my colleague and friend Ed Wilson to famously complain that the study of behavior was largely fractionated, that these two worlds were ongoing but never really interacted. And in some ways, that's why this is such an exciting time to be a behavioral biologist, because I think that these sort of traditional fields are starting to disappear, and now there's a lot of crosstalk between these two fields, which enables us to work at the interface and use approaches from both of these types of fields. And this is really where the work that we're doing in our lab rests. That is, we're trying to understand the molecular basis or the genes, or in fact the precise DNA base pair changes that give rise to increases in fitness. Again, that affects the ability of these organisms to survive, to reproduce in the wild. And once we're able to make this connection between genes and behavior, I think this is where we can really start to address some fundamental questions in the field. This is not meant to be sort of a complete list, but I just want to give you a sense of the types of questions that we're interested in addressing. So for example, this is an age-old question. What are the relative contributions of genetic and environmental effects to behavioral differences that we observe, not just in the lab, but that we observe in nature? And if there is a genetic component, what are in fact the precise DNA base pair changes? What kinds of genes influence behavior? And it's not just that we want to know what the genes are, we want to know how they work through our neural circuitry to actually produce variation in behavior. So to address these questions, what I see as a fundamental challenge in biology is to make the connection between genes, neural circuits, and behavior. So this is going to be the subject of today's lecture. But one could argue that if these questions, again, are not new, why don't we already have lots of examples of genes that affect behavior? This is clearly an interesting subject. Well, it turns out that this is really hard, and it's hard for a number of reasons. So just to contrast to earlier studies in our lab group where we're looking at pigmentation differences, behavior relative to morphology, we tend to think of them, behaviors as having lower heritability. In other words, the genetic component may be a little bit smaller. And that's because, unlike, let's say, a pigmentation trait, a lot of behaviors can have a learned component, or they could be transmitted culturally from generation to generation without any changes in the DNA at all. There can also be a major environmental effects, and not just sort of the environment in which, at the time in which a particular organism is performing a behavior, but it could be even earlier on in their sort of childhood or mousehood. Some sort of environmental experiences can affect their adult behavior. And we tend to just intuitively think of behavior as maybe being more genetically complex. We think of behavior as sort of being a complex trait, so maybe what we're looking for is not genes that have big effects on behaviors, but maybe many genes that each together have a small effect, but then can produce a large change in behavior. And then there's the problem of tractability. So if we want to do this not just in laboratory models, but we want to extend it to organisms that do cool behaviors out in the wild, we have to think about things like tractability. So if you were interested in elephant behavior, for example, you might have a problem with sample size. But of course, some things like having molecular resources like a genome sequence, that problem is becoming minimized in these days. But I would argue the biggest sort of challenge to studying behavior, or at least a big one, is the simple fact that it's hard to measure. So we could take a mouse that has dark pigment and a mouse that has light pigment, and there are lots of very straightforward ways to quantify those differences in that morphological trait. But if we think about behavior, one just has to turn to what is arguably one of the most stereotyped and well-studied behaviors, and that is the behavior of Drosophila, these sort of beautiful Drosophila fruit flies, and that is their mating behavior. So you'll right away be able to tell I'm not a Drosophila geneticist, but this is how I understand their mating sort of ritual to go. So a male and a female will sort of orient towards each other. There's a lot of tapping involved. The male does some waving of its wings and singing. There's some licking, in fact. All of that happens even before there's an attempt at copulation. So you just have to try to now think about if you were going to try to quantify these differences between maybe it's two individuals, maybe it's two different species. Is it the angle at which they orient? Is it how many times they tap? Is it how vigorous that male shakes its wing? How do you quantify the differences in a reliable, repeatable way, and which differences actually matter in terms of the ultimate reproductive success? So one thing that we were trying to do is to try to circumvent this problem of measuring behavior, so we used a little trick. And that is, we've sort of taken this idea from somebody that many of you probably know and have heard of, and that is Richard Dawkins, who maybe in his second most famous book, The Extended Phenotype, that was written in 1989, he put forth this idea of the extended phenotype. And the idea of the extended phenotype is actually one that's quite simple, and that is if genes control a behavior, and that behavior actually produces an artifact, and I'll give you some examples in a minute, just like any other phenotypic product, so for example, the length of our femur, the color of our hair, we can dissect that genetically, okay? So let me give you some examples of extended phenotypes just to bring this idea to life. So one of my favorite is the bowers that are built by Australian bowerbirds. These bowers are made in sort of a characteristic size and shape, decorated in a characteristic way. This particular male bowerbird is building this bower, which he'll walk back and forth in to attract a female. This particular species tends to decorate this bower with blue stuff that it finds in the environment. But what's really interesting is that the size and shape of this is very stereotyped within the species, but can vary dramatically between species, both in the material used, how it's decorated, and the shape of the bower. The same can be said for the nests of swallows. The material that's used, the location of these swallow nests, and the size and shape of these nests can vary dramatically between species. And the same can be said for spider webs. And then this fourth example is one that's a little bit more complex. It's the mound of a termite. It's more complex in the sense it's not built by a single individual, but a whole colony. So the idea is that if genes control the behaviors that result in the production of a bower, for example, of a certain size and shape and decorated in a certain way, then we can treat this bower just like we would treat the length of a mouse's tail in terms of a trait to then look at genetically. And in doing so, we can circumvent the issue of trying to measure the behavior itself, because now what we have is the morphological output of behavior. So I'm going to use this idea of the extended phenotype to tell you a story about connecting genes to behavior. So there are lots of examples of extended phenotype. The one that we've chosen to focus on is burrowing behavior. Now, burrows are produced by a wide range of organisms. Here I'm just showing you a few examples of species that burrow, and then the more or less realistic depictions of those burrows that they make. But you can see things from bivalves all the way up to these complex burrows that are built by prairie dogs. All of these species have independently evolved burrowing behavior. And because it's occurred repeatedly over evolutionary time, that suggests or is indicative of it having a function or functional significance. So why could burrows be important? So we can all think of examples. For example, they can be important to avoid predators. They can be important in terms of thermal regulation, either protection from the cold or from the heat. For those species that meet and mate underground, they can be important for social interactions. They can be used for food, storage, and growth. Okay? So clearly, burrows can be important for survival and then ultimately reproduction. But the other reason we decided to study burrowing is because of an elegant set of studies that was done by a colleague, Carol Lynch, at the University of Colorado, where she used laboratory mice, where she could control the genetics of these mice, and did these experiments in controlled laboratory conditions. So what she would do is take lab mice, put them in these large boxes filled with dirt, and let them burrow. This is a particular burrow that has one entrance, as indicated by that circle, and the outline of the burrow is shown in the dashed line. And what she was able to show is that some components of the burrowing of these particular mice seem to be controlled by genes. So here we had two things that we were interested in, some ecological significance and a hint that maybe genes were involved in burrowing. And just as sort of a side note, because I think this is a fun fact, burrows are one of the few behaviors that actually fossilize. So this is a classic corkscrew-shaped burrow built by a now extinct giant beaver. This particular specimen comes from South Dakota. But there's some hope that potentially we can even go back in time using fossils. So now that we've picked an extended phenotype, the next thing we wanted to do was choose a study species. We're not studying giant extinct beavers, I'm sorry to say, but we've chosen to focus on this particularly adorable group of, I would like to call them charismatic mini fauna. These are the most abundant mammals in North America. I think some of you are probably very familiar with them because they do invade houses, especially in rural areas. But these are peromyscus mice. That's the genus. They're called deer mice commonly. And while to many of you these may look like a typical mouse, they're in fact actually quite diverged from mouse and rat or laboratory models. So even though they're about the size of a laboratory mouse, and I've actually brought one to show you, they're actually outside of this split. So they're about 25 million years diverged from rat and mouse, and these two are about 10 million years. So, like laboratory mice, we can bring them into captivity. We can breed them in controlled laboratory environments and do controlled tests on their behavior. And like laboratory mice, our group and others are building a whole set of molecular and genetic resources. So for example, we have a genome sequence for these mice. But importantly, unlike laboratory mice, in general, these mice are not commensal. And they're really found through a wide range of habitats. So we could go out probably just a mile away from campus, maybe even on campus, set some traps, and we would get peromyscus. But really, you could go to the top of the Rockies, the coast of Maine, the Catalina Islands off the coast of California, and you'd always be able to catch some peromyscus. And because they live in desert environments and forests and so forth, there's a lot of opportunity for local adaptation. In other words, there's a lot of both genetic and phenotypic diversity among these populations. Okay? There are about 55 species of these particular mice, but we're focusing on one of them, and that is this one here called Peromyscus polyanotus. Peromyscus polyanotus is often called the old field mouse because it occupies old fields. So, very clever naming. So, but they do really stick to this characteristic habitat that is open habitat, like these two images. And once there's sort of secondary growth that comes in, you will not find these mice. So our field sites include these beautiful white sand beaches off the coast of Florida, which is no accident. We chose that. The best time to go is around February. But some of our field sites, I have to admit, are a little bit less glamorous. This is a burnt peanut field in Alabama, where we also catch these mice. And the reason that we focused on this particular species is because of this fellow here. This is Francis Sumner, sort of the classic natural historian. He was associated with the Natural History Museum, in fact, a curator. He's shown here, this is a picture from the 1920s, in his field regalia. I have to admit, we do not dress this nicely now in the field or elsewhere. But you can see his sort of dapper attire. And Francis Sumner really is the father of Peromyscus biology in the sense that he spent a vast majority of his career driving around the US catching mice. And in particular, catching mice and describing variation in their morphology, like pigmentation, but also some behaviors. And really, if you go back and read these papers from the 1920s, he's asking the same questions we are today. What's the genetic basis of this variation that we see? How is it generated? How is it maintained? But of course, he wasn't armed with the molecular tools that we are today. So in particular, he had a paper that he published in 1929 with his field assistant that described what I would say is a very unique burrowing behavior of this particular species. And that is their burrows look something like this. So the burrows are characterized by this open hole, a long entrance tunnel, a nest chamber, and then a secondary tunnel that radiates up near the surface, but importantly doesn't penetrate the surface. You can find these burrows out in the wild because you can find this hole, and if it's an active burrow, there'll be this plume of freshly excavated soil that indicates the mouse is sort of actively burrowing in there. So next what I want to do is tell you a little bit about how we catch these mice in the field to illustrate two important points about these burrows. So here's one of these open fields in Florida in February, and here's a hole that we identified, this plume of freshly excavated soil. So we found our active burrow. The next thing we do is we get a graduate student to lie down in the dirt, face first, head near the hole. And what this particular graduate student is doing is taking some plastic tubing and weaving it down that hole till he hits the nest chamber. And once he hits the nest chamber, he blows into that tubing and agitates the mice that are in there. And while we look like we're sort of standing around cheering him on, we have a purpose, and that is the mice are going to come jumping out of that secondary tunnel. We jump on them with the ultimate goal of catching that mouse or mice that come out. And so this little story is to illustrate two things. First, that secondary tunnel that I pointed out is in fact used as an escape hatch. That is, even though we can't see the hole, remember I told you that secondary tunnel comes up towards the surface, but we don't see it. In fact, if we can see the secondary hole, we never find a mouse in that burrow. They seem to abandon it. So they have this secondary tunnel. And then secondly, the size and shape of these burrows are so stereotyped that even though we can't see where they're going to come out, we can predict with very high accuracy where that hole is going to be, such that even sort of slow old professors can jump on these very fast mice most of the time. Okay? So in fact, this burrow is really nicely designed by these mice to be very effective for avoiding predators. And this makes maybe even more sense if I tell you one of their major predators. Sorry, I should have warned you, graphic photo. One of their major predators are snakes. So you can imagine, just like our tubing coming in that entrance tunnel, a snake coming in, it's nice to have a place to run out. Okay, so clearly these burrows are complex, very interesting. probably have a major fitness component for these mice. But the other reason we're studying this particular behavior is because Wally Dawson, back in the 80s, early 80s, did a series of experiments. He actually brought these mice into the lab and showed that they would recapitulate their burrowing behavior in a controlled laboratory environment. And even though his sample sizes were very small, he hinted that they may in fact have a strong genetic component as well. So the first thing we wanted to do was see if we could recreate these experiments. So here on campus, we have our behavior room, and what you're seeing here are 10 large boxes. In each of these boxes, in our controlled environment, so we can control temperature and lighting and so forth, each of these we refer to as our phenodomes, because in these phenodomes, we're going to measure the behavioral phenotype, their behavior. We put a single mouse into this box that you can see is quite large. It's filled with about 1.5 tons of dirt, and we give them this nice slope because it tends to elicit them to produce a burrow in the center of that slope as opposed to hiding out in the corners. So we leave them in these boxes for about 2 days, completely alone, and then we come back after 2 days, trap the mouse out, and now we're going to measure its burrow. And we do this using what we refer to as phenofoam. So phenofoam is the type of stuff that if you have a hole in your wall, you go to Home Depot, you buy it and you squirt it in, it fills the hole, it expands, and then it hardens. So what we can do is actually make a cast of these burrows, and thereby have a physical representation of the behavior that that mouse was doing over the last 2 days. So next I want to show you a video of how we do this. Just like the field, we put our tubing down, and out pops the mice. You had no idea that hole was there, right? Just like a predator wouldn't know. We trap the mouse out of the box, and now we're using that same sort of approach to fill this in with phenofoam. You'll see it comes squirting out that hole. It indicates it's continuous. We now let that harden for a couple of hours. And here's where having lots of eager Harvard undergraduates come in really helpfully, in the sense that they come in and literally get their hands dirty by digging out these casts. And I actually brought, the risk of sort of being, sorry.
That'll be a little bit better.
Okay. So this is one of the casts that's produced by one of the mice. You can see here's that nest chamber, and on this side, in fact, there's nesting material. And then these are sort of the blobs that happen as that comes out of the dirt. But here's an actual sample, one of our data points from one of our experiments, which is 1 mouse over 2 nights. So we literally have mountains of data. Okay. So what I want to tell you about is some very simple measurements of burrows. So today I'm going to talk about the total length of the tunnel, the length of the entrance tunnel, which is just from the entrance to the nest chamber, and then I'm going to refer to this as our escape tunnel and tell you about its presence or absence. Okay? There are lots of things we can do to measure this in more complicated ways, but today we're going to keep it really simple. So the first thing when we started this sort of grand experiment almost a decade ago was to get mice, bring them into the lab, and ask, will they still burrow? This is now, it's almost 30 years after Wally Dawson did his first experiments. So we can essentially take mice that have, Wally Dawson's mice, in fact, that have been in captivity for an additional 30 years, meaning that they've never seen dirt. Their parents have never seen dirt, and somewhere up to 180 generations they haven't seen dirt, and ask, will they still produce a burrow? So here's one of the first mice we used. We dropped it into this big box of dirt, and immediately it started digging. So it's clear then that this behavior is not strictly learned. And even after being in captivity, some of the strains upwards of 180 generations, they still build these characteristic burrows. Now, you could ask about, is this learned? Do they get better over time? So we took some mice and tested them multiple times. And what we found was, in fact, that they do get a little bit better. but they don't get a lot better, at least not statistically better. So here's some early data. Here's just length of the burrow. Here's trial number 1, 2, and 3. And you can see the first trial, they don't build as long as a burrow as the third trial. And if we continued this on, it would sort of plateau off. But this could be either that they're sort of learning or perfecting their skills, or it could be this is the first time they haven't been in a mouse cage and they're sort of freaked out. So to sort of control for the freak out effect, what I'm going to tell you about is data that comes from the best burrow, and usually that's the third trial of the 3. The second thing we wanted to know is there a difference between males and females? In fact, Wally Dawson, when he did his experiments, used only pregnant females, thinking they would be the most motivated to dig. So needless to say, there were a lot of bets going on in the lab. Would the males build burrows, and would they be better than the female burrows? Well, it turns out that their burrows are identical. So it doesn't matter if you're male or female. It doesn't seem to matter if you're pregnant or not. You build long burrows. So I'm going to lump the sexes together. So next what I want to do is tell you a little bit about what we learned from looking at burrows in the field, and then I'll move on to tell you about what we learned by looking at controlled laboratory situations. So as I mentioned, you can go out to this. This is our beautiful burnt peanut field in Alabama. Here's our burrow. We sent, again, graduate students who came back very strong out into the field. And here, for those of you who may be more molecularly oriented, this is a 15 mil conical. It's about this big in size. So you can see some of these mice build very large burrows. And in fact, we had to do 2 castings of this particular burrow. But what we were interested in asking is if we go throughout the range of these mice, do the burrows all look the same? And in particular, in their range in the southeastern US, they live in some places, as I mentioned, these beautiful sandy beaches. And then our burnt peanut field site is in fact hard-packed sort of clay. So if they build burrows in sand, do they build burrows that are bigger because maybe that substrate is easier to dig in? Well, it turns out the first observation from digging up multiple burrows at each of these sites across its range, and sites in particular that have different soil concentrations or compositions, that burrow shape is conserved, meaning in all of these sites, they all built burrows with this classic entrance tunnel and escape tunnel. But there was an effect on some aspects of burrowing. That is, the depth correlated negatively with silt. In other words, if you were in a high clay environment, the mice stopped trying to dig down, and so the depth was less. But interestingly, the length was the same. So in other words, what changed was not the length. Length was statistically the same in all of these habitats, but the depth varied. So in the sand, they'd be burrows that looked like this. I wish I was more flexible because in the hard-packed clay, right, the burrows had a more shallow angle. Okay? And this gave us some sort of clue that these mice actually may be measuring the length of the burrow. But you may have already noticed that the wild burrows, I just showed you a picture of, look at least much longer than the burrow cast I showed you from the lab. And in fact, this was really interesting because, in fact, they are about twice as long in the field as they are in the lab. And this could be a number of reasons. In the lab, we just give them a sort of fixed area. We only give them 2 days. Maybe this had an effect. But there's one more thing I haven't told you about this particular species, and that is these guys, in addition to be very cute, are also one of the few mammalian species that's both genetically and socially monogamous. Okay? So when we jump on these mice as they come flying out of this escape tunnel, it's usually not a single mouse, but often it's a male and a female pair, and sometimes with pups. So the question is, maybe they're longer because males and females are building burrows together in the field, whereas in our experiments in the lab, we've always focused on a single individual in a burrowing box. So of course, the next question we had was simply, what happens if we put 2 mice in one of our burrowing boxes at one time? So will they each dig their own independent burrows in this box, or will they cooperate? And let me say, I use this word cooperation very loosely. So you can decide for yourself if you think that this is cooperation. So in this particular pair, this is two siblings, a male and a female. And when we came back in the morning after leaving them in this box, actually for 2 days, they both were in a single burrow that they presumably built together. So next what we wanted to know is, are these burrows longer when we put in pairs? And we could do this experiment in lots of ways. We could put in male-male pairs, female-female pairs, opposite sex pairs, etc. And that's exactly what we did, and compare them to the length of a burrow that a mouse would build on its own. And it turns out that individuals build burrows that are a little bit over 40 centimeters. When we put them in same-sex pairs, they build burrows that are longer. But if we put male and female pairs together, they in fact build the longest burrows. And this is particularly interesting because what this means is that these mice are making different decisions about how much effort to put in their burrow building dependent on who their partner is. So if you have an opposite sex partner, a potential mate, maybe you want to put a little bit more effort into building that burrow. So this is the outcome of now putting these mice in a burrow together, leaving them for 2 days. We don't see what they're doing. We can only measure the outcome. But next I want to tell you that we have a way to spy on these mice. I'm going to tell you more about that in a minute. But suffice it to say, we can start to watch what they're doing. And what's really interesting is that when we put in male-female pairs, it turns out that they divide and conquer in a different way, in the sense that they divide the labor in an unequal way. In fact, and we have to be careful here not to anthropomorphize, the males do most of the digging of the actual tunnel, where the female tends to follow that male and clean up after him. And she spends a lot of time digging out and perfecting that nest chamber. Okay? But what's especially interesting, and please note sample sizes here are very small, but when we looked at least one same-sex pair, this was not the case, and in fact they tended to contribute in much more equal ways. So this is an area of research that's ongoing, this sort of idea of how decisions are made based on who your partner is in terms of cooperation in this home building exercise. Okay, so now I've told you a lot about this one particular species. Next, what I want to do is tell you about other species in the group, because the first question we had was, okay, this is a really interesting and exciting behavior, and maybe cooperation has evolved in this case, but what about the other species in this group? Do they all do the same thing? Well, here's where we can turn to some diversity in this group, and we can actually do the same experiments that I described for Polyanotus and a bunch of other Peromyscus species. So the first thing we were interested in is the variation among these species. And here I just want you to focus on the major patterns. Here we're looking at, in green is the frequency of burrowing, how often they built a burrow, and in gray the length of that burrow. And there are a number of things that you should notice right away. First, there's a lot of variation among these species. Some species like Polyanotus dig often and a lot, but other species like Eremachus don't burrow at all. And what's exciting about this actually is it turns out that this really mirrors what we know about their natural history in the wild. Eremachus is a rock mouse. It doesn't dig in the wild, it lives in rocky crevices. So we think we're capturing something about their ecology, their sort of real ecology here. The second thing you'll probably notice is that there's a lot of variation among species. And then the third thing you'll notice is that there seems to be this really strong correlation between how often you dig and how long you dig. Okay? But this just tells us something about the length of the burrows. It doesn't tell us anything about this shape of the burrows. So here are some cartoons. These are not to scale, but give you a sense of the types of burrows that are built. So Polyanotus, in fact, is the only one that built this, what I'll call complex burrow with an escape tunnel, whereas these other ones either dig these short little divots. Let me reach in here. So here's our Polyanotus burrow here, and this is the short little sock-sized burrow built by some of these other species here. And there's this one that builds long burrows, but they don't have that characteristic escape tunnel associated with it. Okay? And this data actually becomes even more interesting if we look at it in the context of the relationships among these species. So you can think of this sort of as a family tree. And what this tells us is that even closely related species can have wildly different burrowing behaviors. And the second thing it tells us is that this escape tunnel is unique and what we call derived. That is, it looks like we're looking at in terms of evolution, the gain of behavioral complexity. And this makes things for us more interesting because there's lots of ways to lose a behavior, right? You could lose the behavior of walking simply by having a mutation in your muscle, for example. But actually, the gain of complexity to us may be more interesting. But what's particularly nice about this data is it highlights how, for example, these two sister species can have very different burrowing behavior, even though they're very closely related. And what this allows us to do now is to take a genetic approach. That is, even though these are good species in the wild, if we put them in the lab, in a cage together, a male and a female, and don't give them any other choice, these two can breed, produce fertile offspring, and we can start to decode the genetics that underlie the differences in these two burrowing behaviors. So for the rest of the talk, I'm going to focus on these two species and try to tell you about how we're trying to identify the genes that are responsible for these differences. So the first thing we wanted to do is to sort of make sure that we think that there's a genetic component to these differences in burrowing behavior. And to do that, we did what's called a cross-fostering experiment. We take maniculatus, these are the short burrowing mice, and Polyanotus, these are their little jelly bean sized pups. And what we can do is take the pups from maniculatus, take them away from their mom, give them a new mom, either from the same species, or cross-foster them with the long burrowing species, and ask, does that have an effect? Does it have an effect who your mom is? If your mom's a big burrowing species, do you somehow learn from her to be a big burrowing mouse as well? Okay. So we could do these experiments and suffice it to say that there's no measurable effect. So for example, for maniculatus, a little burrowing one, if you give the pups a mom from that species, they build short burrows, but if you give them the mom that builds long burrows, they still build short burrows. And similarly, these guys build long burrows independent if their mom is the same species or a different species. So this is a nice sort of control to say, okay, maybe your mom doesn't teach you a lot about burrowing. And that may mean that, in fact, genes are important. So here's where we get to start to do the crosses. So we can take, for example, mums of Polyanotus, cross them to dads of maniculatus. In fact, we do the reverse. We take the dads of Polyanotus and the mums of maniculatus, cross them together, make F1 hybrids. Now they have genes from both parents. And the first thing we want to do is put them in a burrow box and ask, what kind of burrow do they make? So again, there was bets going on in the lab. Money was exchanged. And maybe surprising to us was that these hybrids make perfectly complex burrows, okay, that are indistinguishable from these parents. That is, the length of the burrows of the F1 hybrids are similar to the length of the Polyanotus and different from the short maniculatus ones. And maybe even more sort of stunning is that Polyanotus always builds an escape tunnel, maniculatus never does, and the F1 hybrids always do. Okay? So consistent with this sort of depiction. Okay, so now what we can do is take these F1 hybrids and mate them back to these small digging maniculatus for increasing power, taking the things that are most different, and then looking what those mice do. So let's just focus on the length of the burrows. So maniculatus digs small burrows. This is length of burrow here. Polyanotus digs long burrows. Here's our F1 hybrids. They build Polyanotus-like burrows, so big and long. And then if you look at the bottom, these backcross mice, the next generation, when we mated these with these, first you'll notice there's a continuous distribution. Some are small, some are a little bit long. What this means to me as a geneticist is it's not just a single gene. Maybe not surprising that this complex behavior isn't controlled by one gene, but the fact that we still get mice in the second generation, when we've diluted their DNA with the short burrowing parent once again, they still get long burrowing mice, suggests that maybe the genetic basis is quite simple. So the next thing we wanted to do is not just look at the length of the burrows, but we wanted to know about their size and shape. And you have to forgive me for my ability to draw in PowerPoint, but you get the sort of idea. So these are the types of burrows that we saw on these back cross generation mice. We got little burrows, just like maniculatus. We got big burrows, just like Polyanotus, but something interesting happened. That is, sometimes we got long burrows without an escape tunnel, and other times we got little burrows with an escape tunnel. Okay? So this right away means that you can separate the length, genetically speaking, the length of the burrow to the presence and absence of the escape tunnel. But even more interesting is if we simply count up in that second generation hybrids how many didn't build an escape and how many did build an escape. With real data, it's never this close. This is like dream data. You can't get closer to 1-to-1. And again, what this means to me as a geneticist is that the presence or absence of that escape tunnel at least is consistent with a single gene being involved. Okay? And boy, is it fun to start thinking about what kind of gene and what kind of single gene could be responsible for the production of the escape tunnel. Okay, so now we've got these sort of clues. The next thing, of course, we wanted to know is where in the genome are these genes that are controlling these traits. So don't get bogged down in the details. Suffice it to say we can put lots of markers on lots of chromosomes, ask if there's statistical correlations between the types of burrows they make and the types of markers they have in the genome. And what we found was there are 4 regions, okay, that are associated with tunnel building. But even more interesting, if we ask what these 4 regions in the genome actually are associated with, they're associated with different aspects of the burrowing. So the 3 in blue are associated with the length of the burrow, and the red one is associated with the presence or absence of the escape tunnel. Now, this is perfectly consistent with what I just told you, right? And that was, we saw that continuous distribution, which suggested a handful of genes may be involved in the entrance tunnel length. Here we've noted 3 regions of the genome. And that the presence or absence of the escape tunnel is maybe controlled by a single region, like this one right here. And even more interestingly is that these 2 aspects of burrow building are genetically separated. They seem to be controlled by different regions of the genome. In other words, this tells us something about how maybe a complex behavior could be evolved, and that is, in this case, it could be that it's simply putting different behavioral modules together. For example, a module associated with the length of the burrow, and then you add on to it a module associated with the presence or absence of the escape tunnel. Okay, so now we have these regions of the genome, and now we want to narrow in on genes. So again, don't get caught up in the details, but suffice it to say we focused on one of these peaks, the one that was the biggest and strongest and most reliable and repeatable. Here's our chromosome now turned on its side. We have markers. We have this high association. And what's neat is now we can narrow down on the region. And in this particular region of the Peromyscus genome, there are only about 54 genes, okay? Only. So we're down from, 2,000 or 20,000 to 54. And this particular region actually has a high density of olfactory genes, so genes associated with our ability to smell. We thought, okay, those are probably not our best candidates for burrow building, so let's exclude those for now. So now we're down to about 30 genes. And so we can narrow in on genes that we sort of looked at their functions and where they were expressed. We got excited about things, for example, that were expressed in the brain. And we looked at expression patterns, okay? That is, where are these genes expressed and at what levels, and importantly, are they different between the big burrowing mice and the little burrowing mice? And we found 3 genes that showed a pattern that we sort of got excited about. They were expressed differentially. That is, they produced either different amounts of protein or in different places. And in particular, one gene, the gene in the middle, showed a pattern that we got really excited about in the sense that here's our Polyanotus, the big building species, big burrowing species. This is the expression of the gene. It's high. And it's statistically different from the little burrowing species, which is low. But even more exciting is the hybrids have a pattern that's just like the big burrowing species. And why that's interesting is because the hybrids, just like the big burrowing species, build a big burrow. So here we have this correlation, it's not causation, between the 2. Okay, so let me, so to follow up this experiment, we sort of had to bite the bullet in the sense we could no longer avoid looking at the mouse behavior, but we actually had to measure it. So to do that, we've built what we refer to as sort of the mouse equivalent of an ant farm. So here's a 2-dimensional sort of slice of a burrowing box, and we could spy on the mice and watch what they did at night. So here's a mouse, got down to its nest chamber, it rolls over on its back, starts to dig out that nest chamber, and then ultimately will produce an escape tunnel. So by watching them during their nighttime activity period using infrared cameras, we can measure and quantify their differences. And so we can do that in an automated way. So here's, for example, our mouse that we can track using a computer program. This mouse is sort of dinking around up here in the corner. But here's the burrow that it's building, shown in red. The burrow that the digging it's going to do is shown in blue. And we can actually quantify the amount of digging. We can look at when it digs, how much it digs, where it digs, etc. Okay, this got really exciting until we realized that for every mouse, and remember we do 3 trials, and we're doing hundreds of mice, we had to watch 8 hours of videos. And there are lots of Harvard undergraduates, as I mentioned before, but this is a lot of work. So what we did was we actually came up with a program that measures this. And I can already see, even though this worked earlier, that this video, oh, it is working. Here we go. Okay, so here's a mouse digging. This is sped up 900 times, so you're watching 8 hours in just 30 seconds. But you can see that this particular Polyanotus, the big digging mouse, starts digging right away. It builds its burrow, and what we're tracking here on top is the amount of soil that's getting sort of dumped out of the burrow, which is correlated with what we're tracking on the bottom, which is the visible burrow that's being produced. Okay, so that's a typical Polyanotus, and then we can start to do this also in our little digging Maniculatus mice, and you'll be able to see that these mice, just by eye, have very different behaviors. So this mouse runs around, so the white is the sort of cloud space for the mouse. It runs around, and in fact it has built this little ramp here because it does these little jumps, these little sort of, what are these called? Flips, back flips off the side. It does this for several hours. Don't get stuck, keep going. You can see it's not doing much. Jumps around. And then an hour or 2 before the lights come on, it quickly digs this little burrow. And then that's it. It sits in there for the rest of the night. And in fact, we now know that it sort of anticipates when the lights come on and digs that burrow just before it comes on. So now for each mouse, we have images like this or videos like this, and we can start to quantify the differences in the actual mouse behavior that's responsible for these differences in burrowing. And here are some of the things that we've noticed. First, the first question we could have is maybe they dig at a different rate, that Polyanotus just sort of is faster. Well, in fact, they are a little bit faster. They're about 1.2 times faster than the little diggers, but this only explains about the 10% difference in their overall final product. But as you probably already noticed from the videos themselves, is that there's a big difference in the amount of time they spent digging. In fact, these guys spend significantly more time, and this difference in the amount of time spent digging explains almost all of the difference in burrowing. The other thing you probably noticed from the videos is the timing of their burrowing. Polyanotus, we drop them in these ant farms and they start digging right away for the most part, whereas Maniculatus sort of wait till the lights come on. So we sort of consider these to be our eager beaver mice, and these are our procrastinators. And why these patterns are interesting is that all of them suggest that there's a difference in overall motivation to dig. And this really got us thinking about maybe the big difference between these mice is a motivational difference. So the data that I just showed you suggests that it's really both the time they spend digging and how eager they are to dig is different between these species. With Polyanotus, the big diggers doing it earlier and longer. I also showed you data early on that there's this correlation among species. How often they dig is associated with how long they dig, all sort of consistent with this idea of differences in motivation. And then the last experiment we did was look at, in now development, when they start digging. How old are they when they first start digging their burrow? And it turns out Maniculatus digs its first burrow about 25 days of age, whereas Polyanotus starts digging burrows as early as 17 days of age. And what this suggests is they also start digging earlier in their lifetime. Okay? And in fact, these mice that are precocious are ones that dig longer burrows. So again, this all is consistent with this overall difference in motivation, which leads us to what we call the motivation hypothesis. So now if we go back to thinking about the types of genes that may be involved in these differences in the burrows that are being built, what we really would like is a gene that maybe affects motivation, because it would be consistent with this kind of phenotypic data we've been gathering. So remember I told you we had this great candidate gene, this gene that showed an expression difference in the right sort of direction that we thought, and the F1s had the right pattern. Well, I didn't tell you what that gene was, so let me end by telling you about this gene. This gene is called, we call it chrome 5. It's a muscarinic acetylcholine receptor. Don't worry about the details. We know a little bit about what it does in mice. It's expressed in neurons that have dopamine. Dopamine is sort of that reward feeling, right? And it's expressed in regions of the brain associated with reward circuitry and ones that are associated with motor pattern and control. So the first thing we wanted to do is ask, is it expressed in those same regions in Paramyscus? It is. So the same sort of areas that are involved, but maybe even more interestingly, we know from human studies, genome-wide association studies, that this gene is associated with nicotine addiction and morphine addiction. So our current hypothesis is that these mice are actually addicted to burrowing. Okay, so here's where we are right now with this project. We're sort of left with this question, is this the right gene? So it's going to take us several years now, I would suggest, don't tell any of my graduate students I said that, to sort of functionally validate this. So what we'd like to do is take the little digger, transgenically give it the chrome 5 gene, and see if we can get them to produce a longer burrow. So why, you may be asking, okay, the mice are cute, it's fun to watch their burrows, but why are you studying this, and what is this going to tell us more generally, not even just about evolution, but about ourselves? So let me end by saying that the reason we chose burrowing in part is because it's a natural behavior. So we've been using mouse models for a long time to try to identify behavior genes that may be important in behavioral variations in humans, and certainly that's an area of research that should continue. There are lots of great reasons to study those types of behaviors, but by studying a natural behavior, maybe this is a complementary way to think about behaviors that have sort of naturally evolved in human populations as opposed to sort of mutagenesis screens in laboratory mice. We also study burrowing, not again because we're particularly fond of dirt or burrows, but because they're easy to measure. So we get around this big bottleneck in the field of how to measure behavior. And quite surprisingly, in fact, it turns out that this burrowing behavior, even though it's complex when we look about it, may in fact be genetically simple. But I'd like to argue then that this behavior in these particular mice is potentially going to be a great model for understanding not only what genes are involved, but how these genes influence complex behaviors. So as I mentioned, it's really fun to think about how can changes in just a few genes affect this complex burrow structure. Here I've given you a hint at one that may be involved in motivation/addiction. And maybe more interestingly, that by studying natural populations of mice, we may in fact be more likely to identify genes or alleles that have sort of withstood the test of natural selection. These are alleles in mice that are viable, they're doing well in a natural environment, and hence they may be more likely to be the types of alleles that may be segregating or occurring in other species, including humans. So what our lab is trying to do is take tools and techniques from behavioral ecology, neurobiology, and genetics to try to make these links. So let me end by thanking the people that did the work. So this project started about 10 years ago, literally in my garage with Jesse Weber here. This is a photo from my garage. Brant Peterson is a postdoc that joined him. Those ant farms that I showed you, we call them Brant farms in honor of Brant. And the project is currently being taken over by 3 fantastic graduate students here in the MCZ, Hillary Metz, Zain Ali, and Nicole Bedford. And of course, I need to thank a lot of undergraduates from Harvard, Tufts, Northeastern, wherever we can get them, who helped with our project. And with that, I'm happy to answer any questions.
Thank you very much. Those of you who need to leave, please go out, but Hopi's willing to take questions for the next few minutes if there are any.
And I should mention that if you, afterwards, if you want to come see our cast, and I brought a mouse in dirt for folks to see these incredible diggers in real life. Yes?
This idea of behavior being genetically driven is, I don't know anything about this, but it sounds very fascinating and puzzling. Addiction is something where it's either on or off, it seems like. You're addicted or not. So that, but how do you explain behavior that has to do with tunnels and efforts in time, whole programs, being controlled by genes?
Yep. So the question is, how can we envision things, genes that control sort of this how long you do something, as opposed to whether it may be easier to imagine how a change in, let's say, one gene turns a behavior on or off. But how can you imagine a gene that controls how long you do something that's a much more continuous trait than an on-off trait? And so that's what's kind of fun about having this great candidate gene in the sense that we can start to envision how this may work. And so it could be that these mice, as they're digging, the big diggers, just simply get more reward and are more than motivated to keep going on and going on to some level, whereas the small burrower gets a little reward, it just does it a little bit, but it doesn't last very long. That's sort of a maybe overly simplistic way to think about it, but really we can't answer that question about like what's the mechanism until we nail down this gene and then understand how it works. But that's the exciting part in some sense, is like how do these genes actually produce the variation in behavior? And I don't think we have a very good idea of that, certainly not for a representative number of behaviors. Yep.
This gene, candidate gene, and the short diggers.
So we know that both, sorry. Thank you. So the question is, what do we know about this gene in the small digging species? Is it functional? So as far as we can tell, if we look at the sequence, it's there. It's there in the big species. There are some differences in the protein itself. This is a receptor. We know a lot about its structure function. So our first thing was to say, okay, let's recreate these proteins in cell culture and ask, do they signal at different levels? We did that. We didn't see any difference. Okay, but those kinds of experiments are often tough because you may not be doing it in the right cell type with the right experimental conditions. It could be that there's a difference, but just small difference that is biologically important, not we can measure. But then when we did the expression studies, that suggested that maybe it's not the structure of the protein, it's just how much do you have. So it could be that you could have a difference because you have a good protein, a good signaler, and a weak signaler, or it could be that they signal the same way, but you just have more of one. So we're leaning towards the latter at the moment. Yes.
What effect would epigenetics have on behavior?
Yeah, so epigenetics, what effect does that have on behavior? So we know an increasing amount about epigenetics. So I teach the intro course to freshmen on genetics and evolution, along with three other instructors, and I teach the epigenetics lecture. Last year when I taught this lecture, there was no chapter in the book, and this year all of a sudden there's a chapter, but it's still very small. and the point is that we know some things, but we don't know a lot. We know that it's possible that above the level of the genome, which is what epigenetic means, you can modify genes, for example, by methylating them or putting tags on histones where DNA is wrapped around, that affect the accessibility to those genes and hence their expression. And there are some very nice studies in laboratory mice and rats that suggest, for example, their experience as a pup can affect not only their parenting style when they're older, but also that it can happen. This is a little bit controversial still, but they show that there's different methylation patterns, which changes the expression that may be responsible for that sort of change through adulthood that's not controlled by any changes in the DNA itself, but in the sort of expression of that DNA. Because the studies that we do here really focus on sort of the classic Mendelian inheritance and we're doing crosses, what we're focusing on are sort of the DNA base pair changes themselves. And because we've done these cross-fostering experiments that suggest that maybe learning is at least not playing a huge role in this system, we're not as focused on epigenetics per se, but there's a lot of potential and a lot of excitement about that particular field. Yes?
So I may have misheard you during the presentation, but I thought you said that gene that you had identified as a single gene, that one was responsible for the presence of the escape tunnel versus not.
Okay.
So how does that tie in with the motivation?
Yeah. So I wasn't clear about this. So this particular sort of region of the genome that we went after, because it was the biggest, most repeatable, and the strongest sort of genetic or statistical signature, was one associated with the length of the entrance tunnel, not the presence or absence of the escape tunnel, which in some ways, right, is the kind of most exciting. Like, how does that? It turns out that it's at the end of what we think is a chromosome, and that makes it a little bit trickier to sort of narrow in on the region because it's sort of just hanging out there at the end. We're definitely, that's like the sort of next thing we're doing, or we're sort of actively doing. We just haven't got as far with that. But that's going to be a really fun one.
So
methylation get to have that role in evolution? And do you see methylation in every species you look at?
Okay, so methylation associated with
I mean, the
epigenetics.
on-off- the on-off
Yep. So methylation, methylation associated with epigenetic changes. How do we?
On-off.
Yeah, the on-off of expression of genes. Do we see that in all species, and how could that have evolved? So again, this is a little outside of my area of expertise, but suffice it to say, we don't see it in all species. We know it happens in all mammals that we've looked at so far. But there are certainly, I don't think Drosophila, for example, the other sort of model of the workhorse of genetics, has any methylation. So certainly in some insects, it's not the case. And how could this evolve? I think there are people that are certainly interested in this topic, but it often gets sort of explained as very Lamarckian. So sort of this idea that aren't you sort of changing your, in some sense, your genetic material through the course of a generation that can then be passed on, but without changing any DNA. Doesn't that sound like Lamarck's theory of, the giraffe stretches its neck, it has a longer neck, and then its offspring are going to have longer necks. And so it's often sort of thought of as anti-natural selection or anti-Darwinian evolution, when in fact I think there are very plausible explanations about how this could evolve through, that would make Darwin very happy, very consistent with his theory, and that is if this is a mechanism that increases fitness, and there's some genetic control of the stuff that then leads to the methylation, then if that increases fitness, then that's going to be promoted in the population. And so it's something that I try to be very clear with my students about, is that there's at least nothing that I know of in terms of epigenetics and the way that machinery evolved that's inconsistent with Darwinian evolution.
Maybe one more question.
Okay, so I should let our
I would like to ask
series
a question,
host.
if you could comment on the evolutionary basis for these different tunnel patterns. I mean, what is the role of predators, et cetera, and whether or not a person with a given gene, one of these animals would change if they got into situations where there were more predators.
Yeah, so what are the ecological consequences of having this burrow type, and have we done anything that suggests what's driving this sort of evolution of this complex and certainly energetically costly behavior? So this is an area, again, of active research in our lab. We've actually have students, in part funded by the MCZ, to go out to the field to do experiments in the field to ask, are there differences in, for example, survival associated with the size and shape of your burrows within a population? So for now, all I can say is that we can make up stories, sort of these hand-wavy stories about why this may be important, but we haven't done the sort of elegant field experiments to demonstrate that if you produce a longer burrow, that actually translates directly into increased survivability to reproduction. And that's something that we really want to do, to fill out the ecological side. Because as I mentioned, the goal of our lab is sort of to go from ecology down to genes and then back up to ecology again. We just are missing the ecology a bit.
So let me first thank the audience for coming out in force, and thank Hopi for a great talk.